Hello, and a warm welcome to Meet the Authors. My name is Jennifer Claudio, and I'm part of the OSDR Science Communications team. And today we're hosting our newest session of this series. It's an opportunity to to hear from and engage with international authors of publications that were enabled by the Open Science Data Repository. So to the audience and all, please know that the session is being recorded and will be posted publicly for other viewers to see, as well as sent out to the registrants. Our format today, we're going to have a brief overview of the publication by each author, be followed by a few questions from me. And then we're actually going to hold on to our audience questions until the end, where you can raise your hands or type into the chat. But we will have audience questions at the very end, so just hang on to those. Today, we feature Dr. Afshin Beheshti of the University of Pittsburgh, Dr. Begum Mathik of the University of South Florida, Dr. Joseph Borg of the University of Malta, and Dr. Jakub Miczkowski of the Medical University of Gdansk. Dr. Beheshti, could you please let us know a little bit about your publication, Space Radiation Damage Rescued by Inhibition of Key Spaceflight Associated MicroRNAs?
Sure, yeah, thanks. Yeah, thanks for having me and on this nice panel. Yeah, so that work in a nutshell, we're looking at countermeasures. Is there potential countermeasures to mitigate? Is there the magic pill to make you safe to go to space? That everyone wants to have that magic pill. But specifically in that paper, we're looking at microRNAs. That's one of the things I've been studying, and microRNAs are a form of non-coding RNA that the Nobel Prize was one discovery of it was one on last year, as everyone probably heard about. And these microRNAs don't transcribe into proteins, right? There's one form of non-coding RNA, and they, due to their size, they're very stable. They can circulate in your blood, so they're good biomarkers. But also, one microRNA can bind and silence hundreds to thousands of genes, depending on the microRNA. So, in my hypothesis for diseases or most diseases, there should be a microRNA signature that will be associated with that disease. And in this case, I found a microRNA signature from previous work that was related to spaceflight related uh, health risks, in general. And from these, there's components of microRNAs related to specific health risks. And one of them was related to cardiovascular microvasculature damage and risk due to the spaceflight or the radiation microgravity components. So here's a follow-up from the previous work looking at in-depth why how this might behave. Previously, we had shown beginnings of a paper. If you inhibit these three microRNAs, we actually were able to mitigate the damage caused by space radiation, doing simulated microgravity experiments at Brookhaven National Lab, where you could simulate the galactic cosmic rays. So then by inhibiting these three microRNAs using these things called antagomeres, which is an antagonist sequence to the microRNAs, we were able to mitigate the damage. And here's now we did like an in-depth study about looking at the biology and what was being rescued here. So doing sequencing on these 3D organoid models with microvasculature, and with radiation would collapse them. But then when we applied the antagomeres to them, we were able actually to rescue the damage caused afterwards. So we found that there's more efficient DNA damage repair. For example, these three microRNAs, if they're elevated, they tend to cause binding to DNA repair genes. So then, of course, that would inhibit your ability for your cells to repair the DNA damage done by the radiation. So then, if you're able to silence those genes or those microRNAs, those genes are now can be fully efficient to repair the damage caused, right? So then we also found that like inflammatory factors and things like that were actually rescued by inhibiting these microRNAs because these microRNAs help promote that. And also mitochondrial dysfunction was shown these microRNAs are involved with mitochondrial dysfunction. So you inhibit all that, all the nice things that your body wants to do to repair from a damage caused by radiation or in this case, it's purely radiation effects. We're able to mitigate that damage. So, we didn't prevent the damage from being caused because right now, physically, there's no molecule to prevent like radiation causing a DNA break. If you have that, I mean, you should probably market that and sell that. You become a nice billionaire. But you could actually help improve efficiency of repair, right? So that's the point here. So by mitigating these three microRNAs that are elevated during space radiation impact on your cells or tissues, or specifically here microvasculature, we're able to mitigate speed up the efficiency of repair and cause reduce the inflammation associated, improve mitochondrial dysfunction, and everything else downstream. So in a nutshell, that's what this paper was, large collaborative kind of work from a lot of people, originated from, work that was evolving from my grant, but there's a lot of other people who joined the paper to help out and make it what it was.
Cool. Thank you, Dr. Beheshti. Dr. Mathick, could you please share highlights of your publication, Spaceflight Induces Changes in Gene Expression Profiles Linked to Insulin and Estrogen?
Hi, Jennifer. Thank you for this opportunity, first of all. Yeah, I'm happy to talk about our work as well. I was reading some papers about hormones and in spaceflight, how it affects and change. And I came across with a paper from 2016 looking at insulin levels, IGF-1 and HOMA-IR, which is also an indicator of insulin resistance used for clinical research. And I start to take some questions, reach out to Afshin, and I say, what we can do it here. And then he say, go for it. Let's, work on this. And then we created this group. So we dedicated our time, try to understand insulin, and also the estrogen came into the picture, and I will explain the reason behind it. And we started to look at the NASA OSDR data sets. We utilized, first of all, female rodent data sets, then both data sets which has male and female, so we can look at the sex differences. We also obtained from other collaborators and PI. They were so generous to share their data from JAXA and Inspiration 4. So we have the human side of the project as well. The reason that the story started with insulin, as I said, based on literature, we already had a publication. Then one of those prior papers showed that the fatty liver and lipid accumulation in the liver that when mice went to space. So there was a little bit story behind it. And the other thing, while I was interested in estrogens, is my background is I'm an obstetrician gynecologist, but it wasn't the only reason. So estrogen is also known for its act on, it's not only a reproductive hormone, but it acts on many organs and many tissues, and it's involved in metabolism and immune system as well. And estrogen is an insulin sensitizer, kinda. We know this from the animal studies. When there's an estrogen receptor knockout in both male and female mice liver, then you develop insulin resistance. And then in clinically, we have that information too during the menopausal transition. So that was the reason why two hormones, we can merge the story together, but also there are several other important hormones in our body. So basically, the story started like that. We started to analyze data sets, and to the best of our knowledge, like this was the broadest project looking at diverse data sets and looking at the genes associated with these hormones and in their signaling pathways. So yeah, that's like in a nutshell, like the methodology part. And I will explain more what we find it as well, if we have time.
Great, looking forward to that. Thank you, Dr. Mathick. And Dr. Borg, could you please share with us highlights of your publication, Spatiotemporal Expression and Control of Hemoglobin in Space?
Yes, thank you very much, Jennifer, and thank also the audience for being with us today. Yeah, so as the title says, this article, this research manuscript is about hemoglobin expression. I mean, the background, the history is a bit more colorful than just the publication itself. And this is because myself, my team, and also the lab here in Malta has a very long history in hemoglobin research. So being in Malta and the small island population, we do these screening programs where every single newborn and also the mother-to-be, they're screened for hereditary blood disorders, which includes screening for sickle cell and thalassemia. Largely, most of these screen tests result in normal hemoglobin profiles, but then one would identify these rare variants or occasional hemoglobinopathy, some of which include these very rare hemoglobinopathies that result in elevated levels of fetal blood or fetal hemoglobin. So why is fetal hemoglobin interesting in certain conditions? And this is because, so this is the narrative and the why we did this research in space. So the idea is that subjects or people with sickle cell and thalassemia, usually to survive, they require lifelong treatment with blood transfusions and other medicines. So, and it is not without its ill effects. However, we've come to realize that these same patients and subjects with elevated levels of fetal blood and fetal hemoglobin fare much better and usually do not require blood transfusions as frequent. So that is our motivation to understand better how this fetal blood is brought about. One must appreciate that virtually anyone who's born, so you pass through a cycle of 9 months before birth, and after birth, our very high level of fetal hemoglobin goes down, almost to be repressed, like 1% or less, and this is replaced by the adult hemoglobin. And adult hemoglobin is the predominant globin of our lifetime. Now, in the case of space and astronauts, so first of all, there is this question or challenge that to date, in the year 2025, the complete understanding of the hemoglobin switch from fetal to adult hemoglobin is still not fully understood. So this is a challenge that has persisted for many years. Perhaps as a joke or not, so a few years back, we used to say, who knows in the environment of space what happens to hemoglobin, the switch, fetal blood, adult blood. It was like a joke between our lab. Said, who knows if one day we could access or study astronauts' blood. So it stops there. However, in 2020, I was reading an article published by Afshin and colleagues and Chris Mason on NASA twins. So this was a whole collection in Cell, and this was about DNA profiling, RNA expression profiling, and so forth, including blood analyses. I reached out to Afshin at the time, and then he referred me to Chris Mason, and it's like I reached out, like, I've read the paper, I can see a lot of work on blood. Do you have any information on hemoglobin expression and hemoglobin fractions? And said, we know that hemoglobin in general is perturbed, but we do not know anything about the different fractions of fetal blood, hemoglobin A2, hemoglobin A. Said, but the data is there and we can look at it. So from there, we assembled a small team, including informatics, and we looked at all the genetics of at least 3 cohorts of astronauts, which includes the NASA twins, the JAXA samples from Japan, and SpaceX Inspiration 4. So these were 3 independent astronaut groups. And we could observe consistently, which is documented in the paper, that through analyses we see or we show that there is a preferential shift to the primitive type of erythropoiesis, or there's a preference to the fetal counterpart of hemoglobin. And this was a first, and this was very interesting to observe this, because it's showing that despite being adults, and astronauts are adults, when flown to space, now it can be complex, right? It could be an element of stress erythropoiesis, the epigenetics and the environment, also the space itself, which is increasing radiation and microgravity. But all of this is putting some form of pressure or conditional on the bone marrow, on the blood, that shifts back to the primitive or fetal type of erythropoiesis. Now, this to us was the beginning of an observation, and we are following this actively in current and future missions of astronauts. This time around, instead of the data was already available and we're looking at the globin as a second line of research, we now have experiments planned specifically for globin gene switching in an upcoming mission this same year as well. So this is like an overview. So and the paper showed that yes, so the environment of space apparently shifts or enables a person's own blood or erythropoiesis to preferentially express the fetal, or in inverted commas, repressed or silent genes to be expressed again once more.
That's pretty exciting. Thanks for that overview. Looking forward to asking you and hearing questions about that. Dr. Michkowski, could you please provide us with an overview of your publication, Chromosomal Positioning and Epigenetic Architecture Influence DNA Methylation Patterns Triggered by Galactic Cosmic Radiation?
Sure. First of all, I'm also very happy to be here. In our paper, we focus on a way that DNA architecture, three-dimensional DNA architecture, affects the cellular response to radiation. Because the thing is that the DNA itself is more or less 2 meters long, yet it needs to fit into our nuclei. And the way it can fit, it's because of the epigenetic regulation. And the way the DNA is folded and marked and so on. So we wanted to check whether the irradiation or even different type of irradiation somehow interacts with this epigenetic regulation. And it is particularly interested in terms of space because GCR, galactic cosmic radiation, is formed from different types of particles. And in our study, we've found out that this epigenetic marks, like histone marks, like the way the DNA is folded and actually characterized in terms of charge as well. It interacts with this with the response, and in particular DNA methylation, which is one of the probably mostly recognized epigenetic gene expression regulation, is the change in DNA methylation is related with the energy or linear energy transfer, so to say, which depends on the irradiation type, and it's all connected with the way the DNA is folded and the gene and how the gene expression is regulated.
Great, thank you, Dr. Michkowski. I appreciate all four of your overviews to help orient us into our discussion today of how each of
excuse me, if I may, thank you, Lavorka, for putting all these links to our papers in the chat.
Oh, yes, thanks. But yeah, no, I appreciate how all four of you have really oriented us, have really put forth ideas for us to start thinking about in terms of each other, in terms of each other's research, and to help fill these gaps that are actually existing. And my first question is actually for Dr. Michkowski. So you mentioned the histones and some of the structures. In your paper, I was curious, how did the location of DNA within the nucleus influence how much it was affected by radiation?
Well, so one thing is that when we think about nuclei and DNA in the nuclei, we should think about three-dimensional structure. So there is this outer layer and inner layer, and or layers, so to say. And in our study, we divided this three-dimensional object into 5 different layers, simply to be able to look at the effect of going deeper or to be the outer part of this of the nuclei, and we were able to associate the location, this three-dimensional. This is very important. The three-dimensional location, not just the genomic location connected with the genetic sequence, but the three-dimensional location with the response, with the level of change. For instance, the DNA methylation. According to different types of irradiation, because we study irradiations of different energy levels, and the most interesting thing I guess from this part was that depending on the radiation type, the effect can be even opposite. So it's not that this is always just a mess. This is somehow conditioned mess, and we still don't know how.
I see. Does the location, or are there any specific markers like within the DNA or the methylation factors, does it affect the ability to recover or to stay damaged after radiation exposure?
We would love to know that. So far, we didn't do proper experiments to do this. Our study was done thanks to open data science repository. We done our study on the publicly available data, and right now we're planning follow up on this, so to actually check to answer the question you just asked.
Awesome. Awesome. Looking forward to that. My next question or set of questions is for Dr. Mathick. So your study focused on how insulin and estrogen signaling are affected by the spaceflight, and I guess the body's energy management associated with that. So how can treatments for conditions associated with these hormones, the insulin and estrogen, how can they become tailored to address maybe sex-specific differences?
Thank you for the question. So first of all, sex differences overall are important to understand treatment response in every aspect what we do. They have a significant contribution to clinical conditions and their treatment response as well. So in our study, when we look at there, as I said, they were like in the three different layouts. So when we look at the first of all, only female rodents and their different organs, we saw a different response. And liver was the most impacted organ across the other organ systems that we look. We look at the kidney samples, adrenal gland, muscle tissue, but 5 different muscle types under that. So during that, different diverse tissue set from only females, even we see a difference within the female tissue, how those genes or like estrogen and insulin linked genes like differentially expressed and which is the top organ that affected. So that was the one of the thing. Yes, sex differences are important, but also in the same like sex, like females or males, not every organ, every organ can be impacted differently as well. And the second thing, when we look at the sex differences data, we look at the rodent skin samples, and there was important differences we noticed that may impact the wound healing. One example is certain genes related to collagen. Certain collagen genes was mostly downregulated only in males. So, and then when we look at the human samples, we compare the skin samples again from males and females from the inspiration formation, and we noticed that certain, like I think it was the insulin signaling pathway, it was more differently enriched in dendritic cells, females versus males. And why this is important, because dendritic cells are antigen presenting cells in the skin too. So again, it all tails down both of those hormones, their relation and enriched pathways when the altered gene expressions are linked to either immune system or metabolic changes like fatty liver disease or those conditions too. And I would like to give another example because we were talking about not only estrogen, but also insulin. Insulin resistance has also associated with various clinical pictures. And then when we diagnose them, for example, when it's associated with metabolic syndrome, the diagnostic criteria may be a little bit different based on male and female too. So all overall, your question is that there should be tailored treatment per individual, not only also for sex differences as well. And the future in medicine is heading in that direction as well. And I assume it will be the same for Earth and space, like more tailored, personalized treatment. And of course, taking into consideration of sex differences in both, like, treatment response as well.
Great, thanks. Yeah, I think that's valuable. So I'm glad that this area of research is growing. May I ask a question to Dr. Beheshti? I know from our past interactions with you that microRNAs and mitochondria are pretty big into things you do, but here specifically, how does this research inform us about cellular defense mechanisms under spaceflight conditions?
Yeah, no, thanks. Yeah, so I mean, one thing is microRNAs is I know we discussed in the past, and probably people heard me preach this, where they can be used as a good biomarker to indicate health risks, right? Because they circulate, and then microRNAs target specific pathways and genes. So in this case, let's say, damage done by space radiation, you could say, well, if these microRNA levels are up at this certain level, after you quantify them circulating, for example, there'll be inflammation and mitochondrial damage done to a certain level. So now you should probably think of ways to mitigate that damage. So it could be kind of a health check, right? As you do your you go to your doctor, you have your yearly checkup for cholesterol. This could be, maybe a potential weekly check in space, I would say. And then you could use the proper drugs to mitigate that damage, whether it's these antagomeres, which will have to eventually go through all the human trials, right? Or maybe it's drugs that could target these microRNAs too. There's ways you could do that. So that's one way, yeah, to do that. And for example, for this paper that The data is now, of course, publicly available on OSDR, so anything I do ends up on there. So that's people could mine it now and figure out ways to play around with them themselves. But you could relate that now for the microRNA targets too. For example, there are targets that, as I said, target many pathways and genes. So you could relate, let's say, if as we did in the paper, we looked at inspiration 4 data and NASA twin study data that Joseph talked about too, and Pigum did. You can look at that stuff and say, okay, if these targets are going this level up and down, you potentially could reverse engineer and say these microRNAs can be elevated to that certain level or not. So then, you could say for health risks and stuff, how maybe predict what could happen based on what we're showing in this paper and others have shown, for how microRNA progression, if this stays elevated and these pathways are continuously suppressed, what could happen to someone? Does that answer your question? I think it does. Yeah.
I think so. I mean, thanks for the data that you deposit into OSDR for others to actually look at and to help this field also grow. But I want to go back a little just on the integument. Could you tell just a little more about that? You mentioned so it's a molecule and it's dosed. How does that work? Like what's its method of delivery, or how does that work?
Yeah, so I mean, antagonism is a general term, but antagonism would be the antagonist sequence to the microRNA. So, microRNAs are, let's say, about 20 to 22 nucleotides, or so roughly. They could be shorter, a little shorter, a little longer. So they're very small. So then people have come up with ways to, what you want to ultimately do is, since it's a single strand, you want to bind, something to it, so now it doesn't bind to your genes. So now it's inert. So your body would just flush it out, right? Because you're just some debris in your body that doesn't need it anymore. So then that's how antagomeres are. So some companies make antagonist sequences to these called antagomeres. One of the ones I use is from Ohm Life Tech. I'm not affiliated with them. I'm not promoting them anyway, but they have a thing called antagomere through their delivery system. I mean, through their design, they have these lipids attached to the end of the antagonist sequence to the microRNA, and it kind of lets it, in itself self-transfect, in a sense, right? So then you could give it in your cell culture, you just mix it into your media, and then it'll just find your microRNAs, the targets, and bind to everything that it needs to bind to. Or with mice experiments, which are not in this paper, but in other papers, we've done it where you can just give it like an IP injection or IV injection, and the antagomeres will float till it finds the antagonist sequence it needs to find, the microRNA, right? And it'll bind to those. And if it doesn't find anything, then your body will flush it out because it's not binding to anything, right? So then that's the idea. So that's what the antagomeres are. There are other companies that make other versions of these things, to bind to microRNAs and antagomeres, but these ones have been working very well from this company, so I've been sticking with it. But there's obviously people can design their own, figure out ways to do things, But that's the whole idea. How do you inhibit the microRNAs? So you want to find some kind of way to get something in your body quickly or in your cell culture to do that. Again, you could use we have other AI machine learning tools, not in this paper, but other papers, where we could come up with ways to predict FDA-approved drugs to target microRNAs. So then it's a dirtier version because then you have the side effects from those drugs, but while people are working on maybe getting the human trials and stuff approved for these antagomeres, you could have other drugs potentially doing the same kind of almost same thing,
Cool. Thanks.
Yeah.
Dr. Borg, you mentioned the adult to fetal globin switch in astronauts during space missions in general, but does this actually change the way that oxygen is carried in the blood?
Yes. So first of all, one must remember and appreciate, right, that the fetal hemoglobin molecule has a higher affinity for oxygen when compared to the adult counterpart. So, of course, in so doing, this is so because it is more efficient in retaining and extracting oxygen when it is in a maternal setting, right? When there's a developing fetus or developing newborn. Of course, fetal hemoglobin is also important in those instances or cases where there is low oxygen uptake. So I mean, like people who go to very high altitudes. Possibly this also includes confined spaces and the space station and microgravity. So in so doing, this may reduce the efficiency of oxygen unloading in tissues as well. So it's like a double-edged sword. So on the one hand, it is a way how the body is trying to adapt and expressing more the fetal hemoglobin in order to jealously guard oxygen more, but it is less readily given off to the tissues at large. how this might affect impact in space. I mean, we all know, right, that in the context of space, I mean, there's a lot going on in the human body. I mean, there are shifts, redistribution of fluids, reduced mechanical forces. I mean, there's no gravity. So all of this possibly triggers some form of stress signals or stress factors, which we know that stress triggers the fetal type of erythropoiesis and signals that favor reactivation of fetal hemoglobin. We believe, or it appears, that this is an adaptive form, it is an adaptive mechanism by the body in these situations. I mean, the switch might also act as a temporary adaptation to improve the oxygenation in specific conditions. One needs to conduct more in-depth work, how to say, to really understand the whole picture and how this possibly can affect the short-term space flights and space travel, as opposed to much longer term space travel that includes 3 months, 6 months, or maybe a year. So probably we will see some important changes there as well.
Did I don't know if you already mentioned this, but did you already mention how, or if you know whether the globin shift persists afterwards?
Yes. So okay. So because we have, I wouldn't say conflicting, but we see differences in astronauts that have stayed in space for longer as opposed to those who just went up for 3 to 5 days and come back. It appears that the persistence of fetal blood persists longer in those astronauts who stayed longer in space. Most likely, although we did not study this specifically, but most likely, since the globin genes, as small as they are, and the whole locus itself, I mean, they are tightly and finely controlled, finely tuned. There's a lot of chromatin remodeling that takes place, important transcription factors that switch on or off genes. And I mean, most of these are also impacted by the epigenetic modulation and factors. So the longer that this model becomes adaptive or accustomed to express a particular globin gene, then the longer it will take for that to untangle and unfold. Meaning, when astronauts return from space, we've seen that those who spend less time in space, the fetal hemoglobin dwindles down to normal levels pretty fast, I would say. But those who spend time in space for longer take longer to reach baseline levels of HbF and increase back high levels of HbA. Like I said at the beginning, but the studies were not designed in such a way to study the hemoglobin itself. So we hope to sort of address this question in experiments that deliberately target measurements for hemoglobin as well.
Awesome. So I feel like all four of you together, each of the papers we talked about today are really focusing on space health at the cellular level. I'm curious to get Dr. Michkowski's and Dr. Borg's opinion on how do you foresee new technologies and contributions across the globe shaping your future research?
Okay.
Go ahead.
Yeah,
go ahead, Dr. Michkowski.
Okay. Well, so I think that it will help us understand a lot more, not only in space but also here on Earth, how the cells response, particularly from my perspective, how the gene expression is regulated and how we can maybe mitigate some risk or even design countermeasures. But I mean, the most importantly, I'm sure that this, so to say, space-related needs will open the new ways we can study cellular behavior and gene expression regulation and epigenetic processes, simply because they also provide different different levels in terms of environment for cells. We like microgravity or also also radiation, but even microgravity itself, they will allow us to do things that we cannot do on Earth, or at least not simply do on Earth. So I think it's great. It will be a great opportunity for us.
I fully agree. I think I would like to sort of split or divide this into two aspects. So I like to use the words space for Earth applications and space for space. So space for Earth, I mean the kind of research we do, like Jakub said. So we hope to see that the results that emerge can be potentially applied to earthly conditions, right? So if we find or identify an important or a key biomarker that is responsible for fetal hemoglobin reduction, for instance, right? So if this biomarker can be exploited or targeted here on Earth in patients with sickle cell or thalassemia, then potentially we could say that we have achieved or arrived at a therapeutic, at a target that elevates high levels of HbF with the use of space tech or space technology. I always like to see the use of space as an add-on or an added pillar to what we already do here on Earth. Sometimes some people might think that we do space research and we do only space research only, right? But it cannot be more false than that. I mean, we have huge amounts of experiments, data sets, all the work that we do in our labs, which is ground-based, and the element of space supplements and really gives that critical angle to interpret perhaps better the data. That and the second aspect, how I believe all of this will shape in the near future or now, is that because it appears, right, that space travel is becoming more and more frequent, and not just professional astronauts, but also possibly like some form of space tourists, right? And for the time being, this is being confined to low Earth orbit, possibly the space station, which we've seen already, eventually even further away from the space station as well, who knows? And because humans are life forms and we are made up of blood and organs and all sorts of important pathways and mechanisms taking place inside. I mean, it would be interesting to have on-board, on-site, point-of-care testing, blood tests, that instead of the person having to wait and come back to Earth to observe or identify what is wrong, the sort of molecular diagnosis or molecular interpretation of a person can be done in space. I think this is a no-brainer. It will happen. We cannot stay or keep on depending on Earth-based platforms or research or diagnostics, but things have to go to space and be utilized in real time on site. And I think our research combined in some way feeds on what kind of tools, equipment, or tests need to be done to safeguard or look at the safety of humans in space. That's how I see it.
Okay, thanks. I think you raise a super important point about space for Earth and space for space, and how you just mentioned the collaborations that are really happening, and that perfectly leads me to my next question. Leads me to my next question. As researchers, and someone actually touched upon this even in the chat, where do you anticipate seeing future or current interactions between your works? And so if we could get the opinions of Dr. Beheshti first and Dr. Mathic, I'd love to hear from you about that. So where do you see the intersections between your works?
Yeah, no, thanks. I mean, obviously I work with all three of them, so that's easy intersection. But no, but I mean, with the microRNAs, it's obvious, well, not obvious, but for me, one thing that I like to do is find, microRNA signatures associated with all these types of, like, the hemoglobin is going to have, already Joseph put in his paper too, some microRNAs that are related in there. So there are microRNAs there. It might not be a component of my total microRNA signature, but it might be something a little different, right? Because depending on the disease or the insult of injury to your body, due for that certain area of health risk, you might have a different signature of microRNAs associated that I've focused on cardiovascular, microvascular risk. Here, like, for example, for female reproductive stuff of the womb, like, she might have a whole different microRNAs there because there are sex-independent microRNAs too, right? And dependent ones. So there's universal damage caused microRNAs could be, not dependent on male or female, but there are specific microRNAs that obviously could impact the female reproductive system that males don't experience. So that could be an intersection there. We just have to figure out which microRNAs they are. So then you could look at all the issues done, with space flight and insulin and estrogen related things that go haywire. But there's a whole slew of microRNAs in females that are associated with that we could probably think about targeting. And obviously with Jacob, there's the epigenetic changes and the other, like the chromatin changes that are there. MicroRNAs interact some strange ways or fun ways with chromosomes and stuff. Where, like, for example, it's a DNA double-strand break. People have seen in the chromosome there's an accumulation of microRNAs that happen right near the region of the break. And then I haven't really followed up on that literature, but a few years ago, looking at that and then trying to figure out why is that accumulation of microRNAs all of a sudden turn up, whereas a break happened in the chromosome area. And then I could relate with, microRNAs can also regulate like epigenetic changes that happen too, and vice versa. Methylation can impact microRNA changes, so they have this bidirectional thing. So there's a lot of unknowns and things you can explore. We just need funding. So if there's any funders out there, they should give us money. But other than that, yeah, there's a lot of neat intersection we can do because I think, microRNAs are a good, easy target because there's a stability. They could be found circulating if you find the right signature associated with any of these topics that we talked about and more.
Yeah, I agree with all authors. And I think I will start with the insulin estrogen again. And it's not like sometimes people are asking, it doesn't need to be the serum levels of the hormones, but also like it may take time like to see the effect of the molecular changes pertaining to those hormones and their signaling pathways and their action on the tissues. The reason is that if you have a DNA damage like mitochondrial dysfunction or oxygenation, poor oxygenation, so you have something at the cellular level where those hormones act on those organs, then you may have the upstream unopposed effects of it, or it could be vice versa. Because one of the common questions I'm getting at, like, do you have the estrogen hormone or insulin hormone? Not in this data, but it could be normal, but you can still see the prints of insulin resistance or something due to different reason. And insulin resistance, it's like a vicious cycle. All of the things we are working and like studying are also complementing each other. So you can have mitochondrial dysfunction leading to insulin resistance, and it could be vice versa. So it's all intertwined as like we all human beings, because our body is complex. It's not only one thing. That's why I think it's the beauty of all those groups and all those researchers and scientists that we work together. Everybody has their separate niche, but at the end, we all overlap because it is how the nature of the body. And I'm sure we are all looking for the same answer, like what are the changes? You put an individual into a different environment. One of the common questions I get, like Joseph mentioned, why space research? I think human is the same human, you just put the human in a different environment. So you try to understand what are the impacts and what are the changes? Then what are the implications of those changes, short-term, long-term, and are they reversible or permanent? And how we are going to monitor this. And Afshin already mentioned that too, and Jacob as well. Like, so we are kind of all talking the similar, like, common points, but looking from it from a different perspective.
Yes. Thank you for sharing all that. Thank you for sharing these perspectives. And thank you for this section. I'd actually like to move into some of the audience questions. And so audience members, feel free to type into the chat for me to read your question out loud, or you can raise your hand so that we can have you unmute and you can ask your own question if you would like. There was one question that I already saw in the chat that I can read aloud. It's addressed to Dr. Beheshti. Your study used a simulated deep space radiation, the 0.5 grays, which may not fully replicate the complexity of the radiation environment astronauts encounter in space, as space radiation includes varying intensities, types of radiation, high energy protons and heavy ions, and cumulative effects over extended durations. Do you think more follow-up studies should be done to explore this aspect?
Oh yeah, absolutely. Yeah. I mean, obviously you put out studies incrementally, right? We can't solve anything with one publication. If you did, that'd be good. You could retire from your one paper, win the Nobel Prize. So some people might. No, but yeah, it's true though. So how we usually, if most I think most people are familiar with it here, but how we usually simulate space radiation on Earth at Brookhaven National Lab, you go there and you can't unfortunately do that doses of small doses over time that astronauts will get in a deep space mission, right? They get a low dose rate over a year, and then we got the half a grade that they mentioned here over that's the cumulative the estimated dose that, let's say, astronaut will get on a round trip to Mars. It's a year and a half trip, right? They might get more, they might get a little less, but approximately that's what the physicists had decided could be a good estimate. So then the simulated galactic cosmic ray beam at Brookhaven is a simplified version. There's a big 30 ion version, 33 ion, but we're doing the 5 beam simulated just due to budgetary constraints and time wise when we're there to simulate the beam. Has protons that do different energies, but in the middle of it is sandwiched now with the heavy ions, like iron particle and then oxygen and silicon and helium. So that's how we simulate. The physicists decided to simulate. Yes, it's not truly the same thing, but there's nothing on Earth you could truly replicate what happens in space. But as human, we have to try to get, not we can't afford to send everything to space all the time. We got to try to simulate as close as we can, and then yes, you got to follow up. And like for example, these microRNAs that initially looked at, we looked at the twin study data, which had microRNA sequencing that was N of 1, but that was present. And then to do these countermeasure experiments, yeah, eventually, like maybe Joseph can figure out a clever way to put his microRNA integramers in his hemoglobin CubeSat, send into space, and he can inhibit it in his experiments to send to space. And they say, like, hey, does it work? You have to do all the follow-ups. So, and to see in different conditions if it works. And then, that's where we need funding to do that. So,
I see Ela Jasra has their hand up. I think you're following up with the question, so go ahead.
Thank you so much, Dr. Bernstein, for answering that question. I have just another question from the paper, since I'm not the expert in this field, but still I'm curious to know that so the radiation exposure for this in this study was a single dose. But when it comes to actual astronaut in the space, they go for the low dose and the chronic exposure to radiation. So do you think that this might create a variability or some variability in terms of findings, what we have in the paper, because it is just like a simulation and that was one time. So if you can comment on this.
Yeah, no, it's a good question. So yeah, I mean, the idea, the hypothesis here is, I think a lot of people do these type of research is that, if it works at this very high dose and you're able to stop the damage done, the lower doses should, in theory, be stopped too. Now, there is, radiation dose response isn't linear, right? There's that old LET, the linear response model, not LET is a linear energy transfer, but there was the linear model of radiation. They thought it was, with increasing dose, you're going to get the same damage done. But now, of course, that's been disproven because at really low doses, we see different biological responses. Sometimes it's not linear, right? Sometimes you even see beneficial responses at the really, really low. This is gamma, low LET radiation, right? Now with space radiation, people have to do these studies to figure that out too. People are doing it, and we see it's not linear either, the damage done. But, the idea is, let's say mitochondrial damage, whether it's that low dose or this higher dose, it happens, to a lesser or higher degree, depending on the dose. So then for mitochondrial damage, we know if we're able to mitigate the damage done at that extreme, like half a gray dose, right? That's the variabilities that you may see at low dose, in theory, should be also mitigated. And same here thing with the microRNAs. When we looked at it in the previous papers, we looked at lower doses, which was mice sent to space. So it got that low dose at the ISS, and they're being regulated the same way. Not as high, which is good. It seemed like it's probably radiation dependent how much dose they get. So then you would have to figure out if they're at a lower dose, then you give the tagameters at that dose treatment to block the amount of microRNAs that are floating around the body. But we still have to do the studies, like you're saying, to figure that out, because there could be at a lower dose different variability that happens that we're not aware of. That's right now hasn't been done. Because the issue is when you go to Brookhaven, it's not even if you did the fractionated dose that people do once a day at the low dose, it still doesn't simulate what happens in space, because you give it one dose of radiation and then you get 24 hours of recovery because no more radiation, right? Well, in space, you're constantly getting that background low dose of radiations, right? So your cells have a different type of behavior happening. So that's the case. Even the people who do fractionated doses, you're getting closer, but on Earth, there's no way you could replicate that unless you have lots of money and you could buy NSRL for a year and constantly put a little dose in there. I mean, the closest people have gone to is Mike Grell, for example. He has a neutron radiation source out in Colorado where you can actually, it's a room, a radiation source comes out, neutron source, and you can just leave mice in there for a month. And there they get that constant low dose of neutrons, for example. But it's only neutrons, it's not these different ones. But that's kind of like the idea too. If you have some way to create this, and that's the only real way to replicate what happens in space to get to your question. But that means, again, more funding to do experiments to really get to what you're getting at here. It's good, yeah.
Thank you so much for answering the question. Thank you.
We had another question about, have any of the data sets included circular RNA? And if not, will circular RNA be included in future experimental designs? So I think Dr. Michkowski provided an answer to this within the chat, but feel free if you'd like to elaborate on it or just go ahead and restate what you had written, please.
Yeah, I'm actually looking for the link because there's a following question running to the access to the data. I believe this circular RNA are available from JAXA studies. I just don't have the link handy. I'm looking for it. Afshin, maybe you remember where it is?
Yeah, I can't remember either, but it's out there.
Yeah, it is. It
Yeah,
is
I know
somewhere there.
exactly.
Yes.
You can find it if you search.
We'll follow up with you.
Yeah.
We'll follow up. Yeah.
But the circular RNA thing is interesting because I mean, if depending on the depth of sequencing they've done on the bulk RNA-seq, or even you could find circular RNA in that data sets if you know what you're looking for. So just like link RNAs, like there's not much on long non-coding RNAs. There should be, but people could do only so much in the field. But I bet you could find like circular RNA transcript levels in bulk RNA-seq if you're looking, and we just have to know what you're looking for, right? Jacob could probably do it. Jacob could do it.
There was an earlier question that I glossed over. I'm sorry about that. Someone pointed out how Dr. Borg and Dr. Mathick, there's an intersection between your papers about the high levels of fetal hemoglobin and how it affects the accuracy of the A1C measures of diabetes. Oops, I lost that question. Where'd it go? Thus, in prolonged space travel, we would need A1C tests that can specifically differentiate the HbF from normal HB.
Sure. This is because in hemoglobin biology and through HPLC, we fractionate, right, hemoglobin. And hemoglobin A1C is it's glycated hemoglobin for the adult hemoglobin. However, it comes out very early on in the HPLC chromatogram next to or very close to the HbF peak. So indeed, I believe with the correct program and chemistry, one is able to separate better and more efficiently the peaks between the two. So that is one. So I think knowing what samples you are putting on the HPLC and knowing that potentially there can be mixing of peaks, I think it calls for very good method optimization. I think that's important, and I'm very interested to see how potentially this field or this application can be used in a point of care testing setting, which could be either space or out in the field. I mean, it could be potentially interesting to have a device that looks at the various forms of hemoglobin in various settings.
And I would like to say one thing about that. A1C is going to reflect the last 3 months of your glucose monitoring and condition. So we use it clinically, yes, but it's not going to reflect the weekly changing or a monthly changing. So it's going to reflect the 3 months of change due to the physiology of the glucosation and hemoglobin itself. The other things, it's for on Earth, but I'm sure it will be for space as well. Hemolysis and hemoglobinopathies may affect the interpretation of A1C and its levels. So there could be like cautions, like, when we assess the A1C, those things I think we should be keeping in mind when we have the data, like, is there any factors affecting it, or we just seeing the space impact on it? And if for insulin resistance, there are other parameters. The gold standard is an invasive method, but there are other things can be looked it up. For example, just the HOMA-IR calculation itself, which was used in prior paper too. But yeah, all those things are again intersecting to each other, and that's what the beauty of these all collaborations coming up and like brainstorming, talking about learning from each other and discussing and sharing our ideas and expertise as well. So I just wanted to add one line about the A1C.
Thank you. And Ela, although I see your hand, we're getting pretty close to the top of the hour, so we can follow up these conversations following, but I would like the panelists to provide us with very brief closing thoughts to share with us. And if we could just maybe start with Dr. Mujkowski, and then Dr. Mathic, then Dr. Borg, and then Dr. Beheshti, that would be great. So just brief closing thoughts for our panelists, things for us to think about, homework for us to do as we close out our thoughts on this panel session.
So I believe that this all this space-related science is not only interested because it's about space, but it's also about the situation or environment with limited resources, where we don't have access to all the equipment, experts, facilities, and stuff, and we still want to somehow understand and interact with external features. I believe this is very important also on Earth, because we have plenty of regions with so-called limited resources and not always access to all the equipment needed. So I believe that this line of research can be very useful, and I hope that this will provide us great answers.
Great, thanks. Dr. Mathic?
I agree with Jacob, and like agree with all the other authors, as I mentioned. It's a great and fascinating area and field to be in, and it has so many unknown questions. And like, reading, coming up with questions, try to find some answers. It's just great, and I highly encourage everyone, whether you're a space fan or like space movies fan or not, because everyone asks, oh, do you like Star Trek, or like, are you a big fan? It's great, but even if not, it's a very welcoming field. We have so many things to discover, so I highly encourage everyone. Doesn't matter your background is science, doctor, clinical medicine, engineer, teacher, doesn't matter. I'm sure everyone will have a question and like curiosity and try to find the answer in this field. So I highly encourage everyone to reach out to the community, get involved, learn more, ask more, brainstorming. That's all. And thank you for this opportunity. And I'm happy to be here. And I'm sure we will all work in our areas of interest and try to promote, these research for space and wherever we go, humankind.
And Dr. Beheshti?
Or maybe
Joseph.
Oh, did I miss one? I'm sorry.
I will be brief. I mean, for all this, there's so much work, right? There's so much work. So I think here we can be reminded about the effort called SOMA, or Space Omics and Medical Atlas, which is a huge collection and repository of all the articles and research conducted by many scientists throughout, including ours. And I think from here onwards, this kind of medical atlas can only keep on being expanded and utilized by people on Earth and possibly also off Earth. So I thank Afshin and Chris Mason for instigating and starting all this.
Yeah, and like I said, everyone said great words. I'll keep it really brief. Thanks to Joseph for, and everyone here promoting space as it goes. But yeah, I think as everyone says, anyone could get involved, and everyone sure the data's free. So go mine it, and then you could approach any of us and anyone else, to work on neat projects and papers. That's the part. And any question, there's a lot of unexplored areas in space. So there's probably a lot of things that you're thinking of that no one's looked at. So you should look at it. And we're all approachable here. I'm talking to the rest of the team here too, but we're all approachable. So you could approach all of us and we could all help each other out.
Great. Thank you, panelists, for sharing your time with us. Thank you, audience, especially those of you who made it live with us to participate with this Meet the Author session. And so we wish you well on your day. Thank you again, panelists. Thank you for the insights, and thank you for the questions that everyone has asked. Be well, and I've displayed the QR code if you'd like to continue some of these conversations or look for more of the opportunities. There's a request to join analysis working groups, and I'll copy that link into the chat for you all. Thank you very much.